Okay, good morning, everyone. Thanks again for joining us. It's a pleasure this morning to introduce one of my senior colleagues at Northwestern, Dr. Rishi Arora. Dr. Arora is professor of medicine and cardiology at Northwestern, where he also serves as the director of experimental EP research for us. He's done a significant amount of both molecular and genetic research in AFib, particularly as it relates to oxidative stress. So he's going to speak to us this morning about some of his work. The title of his talk is Molecular Mechanisms Underlying AFib, Implications for New Gene-Based Therapies. So I'd like to thank him for being here and taking the time out of his schedule to do this, and I will turn it over to him. Thanks, Rishi.
Yeah, thanks for the introduction, Nishant. Appreciate everybody being tuned in at this early hour. Okay, so I will skim through some of the things because I believe most of the audience is people that are very clinically attuned. So I would try to skim through some of the clinical work that most of you are familiar with. So for example, I think most of you are familiar with atrial fibrillation and its disease burden and clinical impact. But suffice it to say, AF is the most common heart rhythm disorder. Affects not more than 3, but most people feel more than 5 to 6 million people in this country. I think most of you know the statistics in terms of the numbers of hospitalized patients, percentage of patients with heart failure, et cetera, that have AFib, high percentage of strokes, lots of symptoms. That's why they come see electrophysiologists. Huge economic impact. So suffice it to say that the successful treatment of AF is an important challenge in cardiovascular medicine. Again, I think most of you are also familiar with the existing therapeutic approaches to treat AFib, both drugs as well as ablative procedures, which obviously have been around for 2 decades now, and that is the surgical MACE procedure. Unfortunately, there are limitations to current therapies, the therapeutic approaches. Drugs are less than 50% efficacious and can cause life-threatening arrhythmias. Ablation, as most of you know, has about 70 to 75% efficacy in paroxysmal AFib, but even there often requires repeat procedures. Ablation success in more advanced stage or persistent AFib or AFib in the setting of structural heart disease is less than 50%. Even at good centers, especially in long-standing persistent AF, that's more than a year. And unfortunately, progressive improvements in ablation catheters, energy sources, etc., over the last 10 to 15 years have not led to a significant increase in ablation success. So this begets the larger question: why are current treatments so ineffective? And I would say most people in the field would agree that one of the major reasons for this suboptimal efficacy of ablation and other treatments is that current drugs and ablation procedures are not targeted at key molecular mechanisms underlying AFib. So having said that, my lab has been studying a whole host of both up and downstream molecular mechanisms for a number of years. For this talk today, vis-a-vis the translational component of what we do, namely gene therapy, I was going to focus on 3 main aspects of our work, namely the autonomic nervous system, our work on pro-fibrotic signaling, and more recently on oxidative stress or oxidative injury. Okay, so let's start with the autonomic nervous system. The autonomic nervous system, as several of you know, has been thought for many years now to play a role in both the genesis and the maintenance of AFib. The classical teaching has been that adrenergic AF occurs in the setting of increased activity or stress in patients with structural heart disease, for example, post-MI, whereas the classic description of vagal AF is that it's more common during sleep or at rest in younger patients with normal hearts. However, we also know, and from studies done about 15 years ago, 20 years ago now, that the vagus nerve is probably the dominant autonomic within the creation of AF substrate. And we also know that during ablation of the pulmonary veins, vagal responses such as bradycardia, asystole, and hypertension are frequently noted. In fact, one of the early studies in the ablation era showed that if you religiously ablate these regions in the atria that demonstrate vagal responses during radiofrequency, you can actually increase success of AF ablation. But that was early, and then since then, this line of ablation has not been pursued. That being said, there are groups, again, as several of you may know, that are trying to elicit vagal responses at different regions of the atrium, and they're trying to target those. This is called GP ablation, but more about that later. So, having told you what I just did about the background as to the role of the autonomic nervous system in AF, the precise contribution of the autonomic nervous system to the electrophysiological substrate for AF is not known still. And therefore, the last several years, we chose to systematically assess the functional and structural characteristics of sympathetic and parasympathetic nerves in the atria. We chose to do this in normal animals as well as in dogs that have structural remodeling secondary to heart failure and dogs with electrical remodeling secondary to rapid atrial pacing. And the reason we chose both these models is because, as you know, AF is a disease of electrical and structural remodeling. And consequently, if you really are to understand the disease state well, you have to be able to understand it in these two complementary models. So this is just some of our very early work where we showed that you just do immunostaining. We and Peng Cheng's group pretty much showed this data at the same time, that nerve trunks in the atria have normal atria in a very peculiar location. They are located epicardially. They are located around blood vessels. Certainly a predilection for more nerve trunks and the posterior left atrium. Obviously, there are a lot of nerve trunks in the fat pads, but about 30% of the nerve trunks that we saw were actually outside the fat pads. And so these are nerve trunks which are composed of sympathetic and parasympathetic nerve fibers. But obviously these fibers, these brown fibers, the parasympathetic fibers, arise in these ganglion cells, these parasympathetic cells, postganglionic parasympathetic cells. So all these fibers that I'm showing you over here actually arise from, as you can see over here, from these GP cells, and then these little blue fibers are postganglionic sympathetic fibers. So as you can see, blue and brown are co-localized, or sympathetic and parasympathetic fibers tend to co-localize. So clearly the two systems travel together anatomically, and obviously that's one reason why they functionally often appear to act together vis-a-vis genesis of atrial and ventricular arrhythmias. We also showed back in the day when we were still starting our work that a large number of the vagal fibers that supply the posterior left atrium and the pulmonary veins are carried by the ligament of Marshall. This was Joseph O'Bonney's work in our lab. He was here, and he painstakingly did these dissections and found that a lot of the nerves that travel in the ligament of Marshall ultimately are the ones that then end up supplying the posterior left atrium and the pulmonary veins. In a related study, we also wanted to understand the functional significance of these nerves, and so we performed extensive, very detailed electrophysiological studies using high-density plaques, where we looked at multiple refractory periods in the pulmonary veins and the related left atrium, and found that vagal-induced refractory period shortening actually correlates very closely, we found, with the spatial distribution of an ionic current called IKACH. Obviously, this was the correlation here was with the protein subunits of IKACH, namely KIR 3.1 and 3.4. And in addition to that, what we discovered was that the distribution of IKACH also appeared to affect electrophysiological responsiveness. So as you can see over here, IKACH, which is the pink stain, or the brown stain, is very homogeneous in the left atrial appendage and very heterogeneous in the posterior left atrium. And that's one of the reasons we felt that refractory period responsiveness and response to vagal stimulation was more homogeneous in the appendage and more heterogeneous in the posterior left atrium. So having studied the normal atria, we then went on to look at autonomic remodeling in a canine model of congestive heart failure. This is a canine model where we performed rapid ventricular pacing at 240 beats a minute for 3 to 4 weeks. This ventricular tachypacing causes a progressive increase in atrial and left ventricular size and a decrease in left ventricular systolic function, and ultimately leads to extensive fibrosis both in the atria and in the pulmonary veins. So this model, what we discovered was that there was significant neural remodeling in the posterior left atrium in heart failure. So again, these are normal nerve bundles, mostly brown, which is sympathetic, but there's a smattering of blue, brown, which is parasympathetic, but there's a smattering of blue in there, which is sympathetic. And what you can see over here, these other three panels over here are sections taken from heart failure dogs. And as you can see over here, these nerve trunks are much, much larger, both the ones with these GP neurons and those without. So there's just a quantification for just about every aspect of nerve bundle size and parasympathetic and sympathetic nerve number and whatever else we could measure, we looked at, everything was up in heart failure. On the physiological side, even though parasympathetic nerves appeared to be increased, we found that strangely, parasympathetic refractory period. So again, what's the canonical thinking in the ventricle? Sympathetic signaling goes up and parasympathetic signaling goes down. That's the canonical teaching of the ventricle. But clearly, as I'm showing you over here, there's very much of excessive parasympathetic nerves in the atrium and heart failure. So something is very different in the heart failure atrium as compared to the ventricle. But interestingly, even though parasympathetic nerves were significantly increased in this model, we found that refractive period responsiveness in response to parasympathetic manipulation was decreased. So interestingly, when we looked at M2 receptors using radioligand binding studies, there was no significant difference. So what I'm really showing you is that the nerves go up significantly in number, but the receptors don't change in number. So if the nerves go up and the receptors are unchanged, you would expect a very dramatic parasympathetic effect. But interestingly, when you do vagal stimulation or you give atropine, parasympathetic responsiveness is blunted in heart failure. And the reason we found that was the case ultimately is because there's an increase in acetylcholinesterase of the activity. It's a compensation that basically chews up all the acetylcholine that's secreted by these nerves, because without this, I think you would permanently be inactive all the time. So it's a very sensible, intelligent compensatory response by the body. And so when we gave an acetylcholinesterase inhibitor, basically physostigmine, we found that we would completely restore vagal stimulation, as well as this, when we applied carbapenem directly to the atria, even without acetylcholinesterase inhibition, we found that the responsiveness was very good. Again, going to show you that this is not a vagal release problem. The problem over here is that the acetylcholinesterase increases. But despite this compensation by an increase in acetylcholinesterase, interestingly, we found that when we gave atropine, even without physostigmine in these animals, we found a significant decrease in AF duration. And that actually did not get any better with double blockade. Clearly goes to show you that even in the presence of increased acetylcholinesterase as a compensation, the increased vagal nerves that we see in this model clearly are contributing to the maintenance of AF. So clearly, the autonomics play a key role. Then in a subsequent study, we also looked at the role of the autonomic nervous system in the creation of AF electrograms. Again, as several of you know, there's a considerable, there's a fair amount of literature that supports the fact that pathophysiological substrate for AF may be reflected in AF electrograms. So in this study, we gave atropine as well as performed double blockade in dogs that had congestive heart failure. We wanted to obviously understand the electrophysiological significance of the anatomic data that I just showed you on the previous slides. And what we found was, again, as you can see over here, clearly there's an organization that occurs in the presence of both atropine and then especially double blockade. And then another study, related study, what we wanted to do was correlate these AF electrogram characteristics with the distribution of autonomic nerve points and nerve fibers. And so what we found was that change in AF complexity that occurs with autonomic blockade actually correlates very nicely with the amount of nerve-rich fat. Again, as I said, the majority of these large nerve points are located in the fat, even if 30% are not. So this is Shannon's entropy. And the change in Shannon's entropy that you see with atropine or with autonomic blockade actually correlates with the amount of nerve-rich fat. And this is another example where AF organization with autonomic blockade is greatest over a region of high nerve concentration. So again, the encircled regions have lots of nerve trunks, as you can see over here. And when we give autonomic blockade, again, just look at these regions. So this dominant frequency before and after double blockade. The regions that show the greatest organizations are also the ones that appear to have a larger number of nerves. So clearly there's a spatial correlation between these remodeled autonomic nerves and the electrophysiological changes that occur on top of those nerves as reflected in the changes that you saw in these AF electrograms. Again, as I said, we also wanted to see what happens in a canine model of rapid atrial pacing, because that's where electrical remodeling is reflected. And so this model, very quickly, what this model entails is we perform rapid atrial pacing at 600 beats a minute for weeks to months. As you'll see over here, and our very own Anna Feniger was actually joint first author of the study. It's a fairly recent study where what we found was that in the rapid atrial pacing model, and we've known about the sympathetics in this model for a long time, that there's an increase in sympathetic nerves, but no one had rigorously looked at parasympathetic nerves before in this model. And no one had done a detailed electrophysiology, anatomic nerve correlation study either in this model, even though we obviously knew that in this model nerves tend to increase, especially sympathetic ones. So when we looked at this from soup to nuts in terms of bundle size, parasympathetic nerves, sympathetic nerves, ganglion cells, the whole 9 yards, what we found was that clearly, as you can see over here, sort of akin to what I showed you in the heart failure model. The bundle sizes tended to increase, normal nerve bundle size. This is obviously a larger nerve bundle. And then we also found these so-called mega bundles after rapid atrial pacing, where these bundles, some of them actually were so large that you actually could even view them under a 5x microscope. Massive, massive neural hypertrophy. A lot of it is quantified over here. We also then looked at the individual components of these nerve trunks, and as you can see over here, both sympathetic and parasympathetic nerve fibers go up. This was sort of known from before, at least at the myocardial level, and then we obviously looked at this at the level of the individual bundles themselves, but obviously the myocardium is supplied ultimately by these same nerve trunks. Just focus your attention. Parasympathetic nerves, this was a totally new finding, go up very, very dramatically in this part. Okay, so having said that, just look over here. When you have an increase in these nerve trunks, it's not unreasonable to presume that since these nerve bundles are the ones that ultimately distribute nerve fibers to the myocardium, that myocardial nerve fiber number is also going to go up. And that's exactly what happens as compared to these normal dogs. As you can see in the myocardium now, the number of parasympathetic and sympathetic nerves goes up very nicely. We also found that when we actually looked at just the spatial inhomogeneity or homogeneity of parasympathetic and sympathetic nerves, interestingly, this is the posterior left atrium free wall, and left atrial appendage. Even though sympathetic fibers were fairly homogeneously distributed, parasympathetic nerve fibers tended to be more heterogeneously distributed in the posterior left atrium as compared to the free wall and as compared to the left atrial appendage. You can kind of glean a little bit of that from here. It's hard to tell, I suppose. But again, the number of nerve fibers in these fairly evenly spaced panels in the appendage is about the same. Just look at the arrows, whereas the number of arrows and the number of fibers that you can see in these fairly evenly spaced panels tends to be more heterogeneous in the posterior left atrium. Ultimately, obviously, as I said, we're electrophysiologists. We want, no matter how good or beautiful these nerves look, we want to see what effect they have in electrophysiology. And so what we found, just looking at baseline AF electrograms, was that electrograms were more organized in the left atrial appendage, where these parasympathetic nerves, as I said, were more homogeneous than the posterior left atrium. And none of you are foreigners to this if you've been to the EP lab and have done any kind of mapping of the left atrium. Again, shown over here, posterior left atrial electrogram as compared to an appendage electrogram. You see this all the time. Electrograms of the posterior left atrium and the rest of the free wall tend to be more chaotic and more disorganized as compared to what you tend to see in the appendage. And we think this is because of nerve distribution, at least in part. That's just some quantification above and below, but this is really the money slide over here. It basically tells you that AF electrograms tend to be more organized in regions where nerve fibers, especially parasympathetic nerve fibers, are more homogeneously distributed. Obviously, the proof of the pudding lies in eating it. So if you're going to, you have to disrupt the system to really make sure that the correlation that we found in the previous slide is real. And so this is the disruption slide where we gave atropine. And the response to atropine we discovered was more homogeneous in the left atrial appendage again. So again, the money slide over here, I know it's a complicated figure, but just focus your attention on this. These are high density flags we placed on the atrium, baseline map in the left atrial appendage, and the posterior left atrium. We gave atropine, and then this is a delta map. We were subtracting the baseline from the atropine pixel. And as you can see over here, if you just look at the delta map for dominant frequency, the delta map is far more heterogeneous or patchy in the posterior left atrium as compared to the left atrial appendage. Same thing happens with organizational index. Again, this just goes to show you that clearly the autonomic innervation is playing a role, the parasympathetic innervation, because in the presence of parasympathetic blockade, the response is much more heterogeneous in the posterior left atrium spatially as compared to the appendage, which is actually consistent with the nerve innervation pattern that we just saw. Okay, and we did not see this with sympathetic double autonomic blockade. Okay, and so what could be a potential mechanism for what might be going on over here? And so this was a very interesting finding. What we found was that nerve growth factor, which the literature has known for many years now, is a major upstream factor that's responsible for nerve sprouting in the atrium and the ventricle. So we know from previous studies, and our own studies reflected the same thing, that the amount of nerve growth factor goes up very significantly in the rapidly phased atrium. That's not new. But interestingly, when we looked at this in a regional fashion, number one, we found interestingly that there was, in a strange way, even though yes, NGF goes up in the fat pads, it also goes up in the myocardium. And so we scratched our heads and we thought about what might be going on over here as to what the source of this NGF might be. And then if you look at this in a regional way, again, what really surprised us was that the amount of nerve growth factor in the atrium was actually highest in the left atrial appendage. So this led us to believe that nerve growth factor is probably coming from the myocardium. The hypothesis was that it's being secreted by atrial myocytes in response to rapid atrial stimulation. So obviously this is a rapid atrial pacing model. But in AFib, our feeling was that the rapidly stimulated myocytes produced nerve growth factor. So we took an atrial cell line called an atrial-1 cell line, and we did the same thing with atrial myocytes as well, the canine atrial myocytes. But when you pace these atrial-1 cells, as you can see over here, faster and faster, the amount of NGF that's secreted goes up. So clearly, this is a pacing-dependent phenomenon, a stimulation or frequency-dependent phenomenon. And what was even more interesting was when we did variable pacing, what we mean by that is you can do the same 6 Hz pacing regularly or irregularly. When we pace more irregularly, you produce less NGF. And this led us to believe that one of the reasons you're probably seeing more NGF secretion in the appendage is because the appendage, as I showed you earlier, is more regular in its electrograms, right? We know that from our own experiences in the GP lab and what have you, so we believe that based on this data, that one of the reasons you have greater secretion of nerve growth factor in the appendage is because myocytes in the left atrial appendage tend to be more regularly stimulated as compared to myocytes in the other regions of the atrium. So this does become a bit of a chicken and an egg story as to what happens first, but this is our working model where what we're proposing is that nerve growth factor is secreted by rapidly stimulated atrial myocytes. What I didn't show you was that in addition to this huge hypertrophy of the ganglion plexi and the nerve trunks in the GPs, we also see a very significant increase in stellate ganglion hypertrophy. That's not new. Peng Shen's group has shown that before, but we found the same thing. So our working model over here is that NGF is secreted by rapidly stimulated atrial myocytes. This leads to retrograde NGF transport to the GPs and the stellate ganglia, leading to neuronal hypertrophy, which in turn then stimulates nerve sprouting in both the atria. So again, this is basically NGF being secreted by different parts of the atrium, more we think from the appendage for the reasons we talked about. But regardless, we know that nerve growth factor can travel retrogradely up nerves. That's something that's been described in the literature before. So we believe that's exactly what's happening over here. It's produced in the atria, goes up to the GPs and the stellate ganglia. These tend to hypertrophy in response to the stimulation. And once these hypertrophy, you have this diffuse increase in sympathetic and parasympathetic nerves throughout the atria, which is exactly what we found. Also tells you that the appendage may not be an innocent player in what we do in the atria. So to summarize this part of the talk, there is pronounced remodeling of the parasympathetic and to a lesser extent the sympathetic nervous system in AF induced by structural and electrical remodeling. Neural remodeling is more pronounced in the posterior left atrium compared to the left atrium. The clinical implication of this is that parasympathetic and sympathetic of the innervation of the atria is a viable therapeutic target in AF, both in the setting of structural and electrical remodeling of the atrium. So then the question that arises then is how do you selectively target parasympathetic signaling in the atrium? So this now is a segue into our gene therapy talk. Again, the reason I took my time getting to it is because, remember, first and foremost, for those of us who know what we do in my lab, we're really an AF mechanism lab, and the gene therapy work is the translational component of our work. So without understanding the mechanistic piece, I think it's pointless going into the gene therapy right away. So this is the autonomic signaling cascade. If you had to target parasympathetic signaling, you could do it a couple of different ways. You could target the M2 receptor, but that's not easy to do. As many of you may know, remember from med school biology, there are 5 types of M2 receptors, and unfortunately, there's a very large, very high sequence homology between these receptors, and so it's very hard to come up with selective M2 receptors. Just very, very difficult pharmacologically. But at the same time, we know from other people's studies in the last several years that if you wanted to target a G protein coupled receptor, you could do it much more selectively by targeting the interface between the GPCR, in this case the M2 receptor, and the downstream G alpha G protein. So we know that the docking of the GPCR with G alpha, with the G alpha protein, in this case G alpha I and O, is an area that's evolutionarily very, very conserved. And so if you wanted to target any GPCR signal, you could do it by going after the interface of the G protein coupled receptor with its cognate G alpha G protein. And so that's what we, and again, this is something that's been described several years ago by a group out of Vanderbilt, where people have shown that G protein inhibitory peptides that target the C terminus of the G alpha subunit, which is the one that interacts with the G protein coupled receptor, can selectively inhibit downstream GPCR signaling. And so this is just one example, but you can do this with other G protein coupled receptors as well. People have done this with the endothelin, for example, G alpha Q, G alpha 11, 12, a lot of different receptors have been targeted by this method. So our hypothesis over here was that targeted disruption of G alpha I and O signaling in the posterior left atrium. So G alpha I and O is downstream of muscarinic signaling. So our hypothesis was that targeted disruption of G alpha I and O signaling in the posterior left atrium using C-terminal inhibitory peptides would lead to selective vagal denervation in the left atrium and a consequent decrease in vagal AF. So what we've done initially, what we did was we put in a mini gene, a small gene with a very small insert that expresses these G alpha I and O inhibitory peptides. And then we looked at the refractory periods and AF inducibility 48 to 72 hours after injection of gene. And so what we found over here is the posterior left atrium is where the gene was injected. And this is just delta ERP in response to vagal stimulation. Vagal stimulation, as expected, in the presence of scrambled or dummy peptide, is very strong. It's like a normal atrium. 40, 50 milliseconds refractory period shortening. If you just give the one gene expressing the one peptide G alpha I2 over here, you will find that there's about a 50% abrogation of vagal induced refractory period shortening. If you give both the peptides in mini gene form, you almost completely obtun vagal induced refractory period shortening. But what's even more interesting over here is that even though we injected the gene only in the posterior left atrium, and not unexpectedly, you see effects of the posterior left atrium, but interestingly, you see an effect also in areas that are remote from that region of injection. So the pulmonary veins, they're not too far, but they are because we're not injecting on top of the pulmonary veins. And yet, you see a response in the pulmonary veins, and even more surprisingly in the appendage, which as electrophysiologists know is really quite distant from the posterior left atrium. And again, we see a very powerful effect in the left atrial appendage as well. So clearly, just injecting the posterior left atrium is affecting vagal-induced refractory period shortening of the whole left atrium. And this is obviously reflected in a decrease in vagal-induced AF, both in the number of episodes as well as the duration, as well as the number of episodes that you induce. And why do we think we get this remote effect of the whole atrium? We think the reason for that is the brown stain over here is a flag stain. So basically, the gene that you inject has an epitope on it, a flag epitope. And so if the gene gets expressed, then you should be able to see the flag epitope as well, which is basically brown. And so as you can see over here, this is normal myocardium, uninjected. There's no brown over here. This is thick brown stain because there's a lot of flag peptide in this myocardium. These are myocardial myocytes. But interestingly, as you can see over here, there's also a good amount of the flag in the nerve trunks. These are nerve cells, and these are GP neurons. So clearly, when we electroporate the atrium at these low energies, gene is taken up not just by the myocytes, but also by the neurons. And I know the word on the street is that when you do high energy electroporation, that it's very selective for tissue. And that's one of the reasons people believe that it might be a better energy source for the future in terms of collateral damage and so on. But I can tell you in our own hands, if we do low energy electroporation, you clearly are getting gene into multiple tissue types. So but that's just our observation. And then obviously, since the goal would be to go longer term with this gene-based approach, we have also performed longer term expression of G-alpha INO inhibitory peptides in a chronic AF model. This is ongoing work in the lab. And what we found is that there's delayed onset of AF after injection of these mini genes expressing G-alpha INO inhibitory peptides. So to do this, we have to be able to clone the insert of interest into a plasmid that can express longer. So we have a plasmid that's called polyubiquitin C plasmid. So it's a polyubiquitin C promoter, and it's a pol-2 promoter that several studies have shown over the years actually expresses much longer. It's not silenced as easily as the promoter that I showed you in the previous slide, which is a CMV promoter. And so when we've used this promoter, we've actually had very good success in terms of long-term expression. But this is just some of our earlier data that goes to show that if you inject this gene of interest, if you don't inject this gene within about a week or so of pacing, as Dr. Penninger will tell you, these dogs begin to go into AFib that begins to sustain more than 30 minutes at a time. But if you inject the G-alpha INO inhibitory peptides together, as you can see in this particular series of 3 or 4 animals, you very significantly delay the time to onset of AFib. So to summarize this part of the talk, these results demonstrate the feasibility of a targeted mini gene-based approach in achieving G-alpha INO inhibition of the posterior left atrium, with a resulting change in vagal responsiveness in the entire left atrium. Non-ablative methods as a result of this data, we believe, can be successfully employed to attain selective parasympathetic inhibition of the left atrium, and appropriately selected ablation sites, we believe, can lead to downstream effects that lead to favorable modification of AF substrate. Okay. Quickly shifting gears now to the next part of the talk, fibrosis. Atrial fibrosis has been thought to be, for years now, to be one of the most important factors in the formation of AF substrate. Atrial fibrosis has been observed in biopsies from patients with AF and people with risk factors for AF, such as advanced stage valvular disease, dominant cardiomyopathy, the list goes on and on. In the heart, we know that TGF-beta-1 is a very important signaling molecule that increases atrial fibrosis, as evidenced by overexpression and knockout models. We know that serum TGF-beta levels increase in patients with AF from early clinical studies, and we also know that when you cardiovert people with AF, you have a significant decline in TGF-beta levels. And so this is, again, fibrosis is complex. There are multiple signaling pathways that are involved in it, but we know that the TGF-beta is an important one, and so this is one that we decided to target. And the reason I'm showing you this slide is because we know the TGF-beta signals by one of 2 important mechanisms. Is the canonical pathway, which is a SMAD related pathway where via phosphorylation of SMAD2 and 3, you turn on pro-fibrotic genes in the nucleus, because the SMAD transported to the nucleus where it turns on these transcription factors that ultimately lead to collagen synthesis. But there's an alternative pathway as well, a TAC1 alternate pathway, which via the MAP kinase pathway can also turn on the same transcription factors in fibrosis. So our hypothesis over here was a decreasing TGF-beta signaling in the canine posterior left atrium will attenuate heart failure-induced fibrosis, heterogeneous conduction, and resulting AFib. How do we accomplish this? We injected a plasmid that expresses a dominant negative type 2 TGF-beta receptor. So the classic TGF-beta receptor, like all receptors, has to signal to the inside of the cell by some mechanism. And so what you're doing with this particular approach is you're overexpressing this truncated receptor that cannot phosphorylate downstream. So basically, it lacks an intracellular kinase domain. And so as a result of that, it's not able to signal. And if you overexpress enough of this, hopefully you'll be able to basically competitively decrease the native normal TGF-beta receptor, and as a result, stop downstream signaling, or at least decrease it. So that was the goal, to competitively inhibit the normal TGF-beta signaling by creating a defective TGF-beta receptor. And so again, this was performed in the canine heart failure model. As I've shown you before, this is rapid ventricular pacing at 240 beats a minute. But the reason I'm showing this to you again is because this model, as I said earlier, produces fibrosis. And this fibrosis leads to an increase in conduction heterogeneity as measured by something called conduction and homogeneity index. And this will come up again. That's why I mentioned it over here. So this is how we performed the gene injection, transgenes under the control of the same promoter that I mentioned earlier, the UBC promoter. So basically the TGF-beta dominant negative R2 receptor was cloned into a UBC backbone. Control plasmid in this case was a LAC-C expressor plasmid. We performed electroporation. So you do a gene injection, it's a subepicardial gene injection. You raise these little blebs all over the epicardium, and then you perform electroporation. These are electroporation electrodes. After gene injection, you perform a few days later. We started ventricular tachypacing at 240 beats a minute for 3 weeks, and then we performed a terminal study to assess conduction and fibrosis, etc. So quickly to the results, what we found was that the dominant negative receptor attenuates conduction and homogeneity in the heart failure inferior posterior left atrium. So again, in controlled animals receiving the LAC-C plasmid from baseline as compared to 3 weeks of ventricular tachypacing, there was a very significant increase in conduction and homogeneity. However, in animals that got the active gene, as you can see over here, at different cycle lengths, pacing cycle lengths, when you go from baseline to post-ventricular tachypacing, there's no significant increase in conduction and homogeneity. It's just an example over here. But interestingly, we found that not only do you improve conduction characteristics, you also positively affect the APD restitution slope. So we looked at MAPs in these atria and using a monophasic action potential probe, and we found that the APD restitution was actually improved in these dogs. So we think it's a dual mechanism. Obviously, ultimately, what are we interested in? We're interested in AFib, and with the dominant negative receptor, there's a very significant decrease in AFib, as you can see here. Again, we're electrophysiologists. Obviously, all aspects of electrophysiology interest us, and so the electrograms are always near and dear to our heart. And as you can see over here, interestingly, this came as a bit of a surprise to us, but if you think about it, it doesn't have to be. If you take away a little bit of the fibrosis, kind of the thinking today is, well, if you have fibrosis, you're going to cause more chaotic or more disorganized AF electrograms. Well, in our case, we found that electrograms were more disorganized in the presence of lots of fibrosis, which is 3 weeks of ventricular tachypacing. And then when you took away some of the fibrosis, obviously you can't remove it completely, you find that electrograms tend to get faster and a little bit more disorganized. So we think what happens is that, yes, electrograms do tend to disorganize electrograms early when you have a little bit of interstitial fibrosis, but once you get lots of fibrosis eventually, and we've seen these atria, several of us have, when you have somebody with structural heart disease, what you see in these atria, or think of the post-ablative atrium or the post-Mays atrium or a heart failure atrium, you see these slower, sick-looking, chronically, low amplitude type electrograms in the atrium. We think that's what chronic severe fibrosis does to you in the long term. A little bit of fibrosis, yes, there's a lot of disorganized activity, but once fibrosis becomes severe, you slow down and basically organize the AF electrogram. So why did all this happen? Why did we see these improvements in AF and conduction? Well, the reason was at the tissue level, an improvement in the amount of fibrosis. So again, this is a control animal, lots of blue or fibrosis you can see over here. This is this is a dog that received the dominant negative receptor. As you can see over here, there's very little fibrosis, much less. So it doesn't go away completely, but there's a lot less. That's quantified over here in multiple animals. Again, with gene therapy, one has to be careful that you get the gene cargo into as many regions as possible. Again, basic electrophysiological concept. If your gene expression is patchy, potentially you can always be pro-arrhythmic for reasons that most electrophysiologists are familiar with. And so in our hands, we find that with the approach that I showed you, the electroporation approach, the sequential injection electroporation approach, gene expression is moderately homogeneous. The green is the gene. It's not completely, but moderately homogeneous. This is the uninjected region. That's a V5 tag. And in terms of the signaling pathway that I mentioned to you there too, we were able to get this improvement in fibrosis and downstream electrophysiology, not only by affecting the canonical SMAD pathway, but also by affecting a MAP kinase phospho-ERK pathway. Last part of the talk, oxidative stress. Oxidative stress is thought to be an important contributor to both electrical remodeling and structural remodeling in AF. Oxidative stress is thought to affect cardiac ion channels and excitation contraction of the coupling, at least in part by increased CAMKII signaling. Unfortunately, most data that supports the role of oxidative stress in AF is indirect, extrapolated from findings of inflammation and oxidative damage in explanted atrial tissue. And the precise mechanisms by which oxidative stress creates a vulnerable substrate for AF and the intact atrium are not known, and neither is it known whether oxidative stress affects emergence of AF triggers in intact pulmonary veins. So this, the next few slides are just going to summarize data from last 4 or 5 years of work in the lab in this space, but really it's a two-part hypothesis. Number one, in heart failure, we believe that there's a preferential increase in oxidative stress and downstream CAMKII signaling in the intact posterior left atrium, which creates a vulnerable substrate for triggered activity and reentry. And the second part of the hypothesis is that oxidative stress contributes to both the initiation and the maintenance of electrical remodeling in AF. So this is some of our data in a heart failure model, which is the first part of the hypothesis, where we think an increase in CAMKII signaling via an increase in ROS leads to conditions for both triggered activity and reentry. Again, in this heart failure canine model, very significant increase in superoxide generation. It appears to be preferential in the posterior left atrium, even though it does go up in the appendage too. So this increase in superoxide then actually leads to an increase in protein oxidation as measured by an assay called carbonylation. So if we just focus over here, for example, you're looking at just the total number of bands over here, the darkness, and you can see it's clearly carbonylation is very significantly increased in the setting of heart failure, right? Quantified here, which is protein oxidation. And again, we found that it was preferential. There was more protein oxidation of the posterior left atrium compared to the appendage. This increase in oxidation is accompanied by an increase in CAMKII phosphorylation of, well, two things happen. There's an increase in oxidation of CAMKII, which is a master signaling molecule that exists in both of the atrium and the ventricle. There's an oxidized form of CAMKII that goes up in the atrium in response to this increase in oxidative stress that I showed you. And this increase in oxidative stress then is accompanied by an increase in expression of CAMKII phosphorylated RYR2. And this particular site of phosphorylation of RYR2, as several of our fellows will be familiar with, is something that has been described before, initially by Andy Marks and then by Zonda Werens, as being a site that actually makes the renin receptor leaky. This is one of the famous calsequestrin mutations. Sites of one of the famous mutations of the calsequestrin mouse, which leads to CPVT. But we know also from Zonda Werens and other people's work, Stan Atell's work, that the same phosphorylation of RYR2 can also lead to leakiness of renin receptors in the setting of AFib. So anyway, bottom line is when you have an increase in ROS, you have an increase in oxidation of CAMKII, which then leads to an increase in CAMKII phosphorylation of RYR2 at this very interesting site. And then at the physiological level, we had shown together with Andy Wasserstrom's group a couple of years ago that when you actually, so there's something called triggered calcium wave, which is a very unique calcium release phenomenon that we see in atrial myocytes. Not in ventricular myocytes. We believe that because T tubules are pretty poorly developed in the atrium as compared to the ventricle, when you pace myocytes faster and faster, it's a calcium transient. At fast atrial pacing, you will not see this in normal ventricles. You begin to see these calcium waves. We call them triggered calcium waves. These are very atrial-specific calcium waves. But the important point for this particular slide is that in heart failure, we found that there's a very significant increase in these triggered calcium waves. So one of the reasons we believe that happens is because there is a very significant breakdown of T tubules that happens in heart failure. So in heart failure, you begin to see these triggered calcium waves at much slower cycle lengths. That's quantified over here. So again, to put everything together, increase in oxidized CAMKII, which leads to an increase in CAMKII phosphorylation of RYR2. And our belief is that this increase in CAMKII phosphorylation of RYR2 is one of the reasons you begin to get this excessive calcium release in the heart failure atrium. So I said before, there's T tubule breakdown, but we think this is another major mechanism by which you might be getting more this ROS-induced increase in CAMKII phosphorylation of ROR2 at this 2814 site. We think that may be one mechanism whereby you might be getting these more triggered calcium waves. So again, the proof of the pudding always lies in eating it. So what we wanted to do was then see whether these triggered calcium waves are actually ROS sensitive. So in the presence of phytotempo or aprocyanine, which are ROS inhibitors, as you can see, these triggered calcium waves tend to get abundant. So again, that I think is very significant proof of the fact that these triggered calcium waves are indeed likely, at least partially, caused by an increase in oxidative stress in the atrium. Obviously, with large animal studies, you know, as I said, with science, it's always about doing the sufficiency experiment is always hard to, you know, yes, it's easy to show that there is, if you take away something, that ROS is contributing to these calcium waves, but if you want to show that ROS is sufficient, then you have to go the other way and increase the ROS and see if you bring back these calcium waves. And that's a little bit harder to do in a chronic model. It's easier in a large animal model, easier than in a mouse. But you can actually compensate for that by doing computational studies, computational modeling studies, and that's what we did over here, where in a computational model that one of our collaborators created with us, what we did was we basically married a couple of different models that already were in existence. There is a model that you can use actually to increase CAMKII that comes out of Tom Hunn's lab at Ohio State, and there's a model that you can use. This is by a model of our collaborator, Johannes Schifferall, where you can actually cause formation of triggered calcium waves in the atrium. And so what Johannes did in this particular case was he basically married these two models so that now in the presence of an increase in CAMKII-induced ROS, the question is, can you then bring on triggered calcium waves? And sure enough, as you can see over here, at low concentrations of oxidative stress, nothing much happens, but as you get more and more ROS into the atrium, the same cycle length of pacing as you can see over here, you go from these sparks to these waves that begin to form, and then you see these very real calcium waves, large calcium waves, microcalcium waves, along the lines of the calcium waves that I've shown you experimentally. So clearly, as you increase ROS in this model, in the setting of increased CAMKII, you can actually lead to an increase in triggered calcium waves. In the same model, we also found that there's a preferential increase in CAMKII phosphorylation of the sodium channel, NAV1.5. Unlike NAV1.5, unlike native NAV1.5, what we found was that this CAMKII phosphorylated NAV1.5. So again, this is work that came out of Peter Moller's lab, where he showed that CAMKII phosphorylation of NAV1.5 at this site. So NAV1.5 is the most important cardiac subunit of the sodium channel, and CAMKII phosphorylation at the NAV1.5 site, we know from Moller group's findings, can actually make the sodium current, can actually change the sodium current so that it actually, in their studies, they find that with the CAMKII phosphorylation of NAV1.5 at this S571 site, there's a very significant increase in late sodium current. That's their finding, but we were curious to see whether the same mutation, so there are really two groups that have done this, or three groups that have done this in a very systematic way. Don Burr's group has actually looked at CAMKII phosphorylation of NAV1.5. He's identified a couple of mutations that lead to late sodium current, but for a variety of different reasons, we're especially interested in this mutation from Peter Moller's lab. So we obtained his antibody, and what we really wanted to see was what happens to the sodium channel per se, just histologically. And so what we found was that there was an increase in CAMKII phosphorylation of NAV1.5 at this site, in this heart failure model. But interestingly, what we found was when we looked at CAMKII phosphorylation, CAMKII phosphorylated NAV1.5, very surprising. So you're expected to always have sodium in two main compartments of the cell, at the intercalated disc, and then also at the lateral membrane. Those are the two main compartments where NAV1.5 is, where sodium current is thought to exist. And again, this is what you're expected to see in control myocytes, but in heart failure, surprisingly, unlike native NAV1.5, it does exist in both compartments. What you see over here is for this CAMKII phosphorylated channel, it only exists in one compartment, which is the intercalated disc. And we know that this is a very interesting, important finding. That the ramifications of this finding are huge because we know that absence of NAV1.5 in the lateral membrane will decrease your peak sodium current by about 40 to 50%. I mean, it's a mass effect. We know that you need sodium current in both compartments. And so if you take the NAV1.5 away from one compartment, we know that peak sodium current will go down. And so based on this, the implication of the work from our perspective was that an increase in this defective variant of NAV1.5, this CAMKII phosphorylated variant, likely contributes to slow conduction of the heart failure for atria. Now, why does this happen? Again, there's some very elegant biology that has been done by a couple of different groups in this area as to why you have two different pools of sodium current. Suffice it to say, we believe that the reason sodium channel exists in different pools is because different binding partners take it there, different parts of the cell membrane. And so anchorin G is one of the key proteins we know, binding partners, that has been known to bind to the NAV1.5 CAMKII phosphorylated NAV1.5. We know that from Muller's group. And so that's one of the first things we want to look at. And then we looked at the amount of anchorin G. We found that the amount of anchorin G actually goes down very nicely in heart failure. So this actually then makes a lot of sense. And this is again, fluorescence, as you can see over here. Even though there's a lot of cadherin at the intercalated disc, there's not much anchorin G. So if you have a decrease in the main binding partner that takes your NAV1.5 to the left to the lateral cell membrane, it stands to reason that may be one of the reasons why not enough NAV1.5 is being transported to the lateral membrane. And so again, this just is a mechanism, is a likely mechanism whereby we think there is an insufficient amount of, or actually a negligible amount of CAMKII phosphorylated NAV1.5 because its main binding partner is missing in heart failure. So what happens as a result of that is that there's patchy dropout of CAMKII phosphorylated 1.5. So again, this is just, again, if you're going to do this in intact hearts, you have to put it all in the context of inhomogeneous conduction and conduction velocity. But suffice it to say that because there's patchy dropout of CAMKII phosphorylated NAV1.5, again, as I said, it barely exists at the lateral membrane, it only exists at the intercalated disc. So as it is, there's a decrease in conduction velocity. We definitely see that in the heart failure model. But then in addition to that, because this knockout is patchy, so interestingly, even the little bit of, in the interest of time, I don't want to belabor this, but even the CAMKII phosphorylated NAV1.5 that is left at the intercalated disc, we know is knocked out in a patchy way. Maybe I did not do justice to this. Anyway, what I want to say is that because of this decrease in anchorin, what happens is that even the CAMKII phosphorylated NAV1.5 that exists at the intercalated disc, I misspoke, anchorin G actually takes the NAV1.5 to the intercalated disc and not to the lateral membrane. Sorry, it's the early hour. And so what we find is that because of a decrease in anchorin, there's a decrease in CAMKII phosphorylated NAV1.5 in a patchy fashion also at the intercalated disc. Anyway, make a long story short, there's very heterogeneous distribution of CAMKII NAV1.5 because of this decrease in anchorin, which is reflected in more inhomogeneous conduction of the posterior atrium. So we again created another computational model with the help of Ivana Shifarov, where what we wanted to see was in the presence of fibrosis alone, which people have for years said is enough to give you inhomogeneous conduction, is that indeed the case? So what we found was that inhomogeneous low conduction, as created by this CAMKII phosphorylated NAV1.5, actually interacts with regions of fibrosis to exacerbate substrate for reentry. So if you just have fibrosis alone in the atrium, you can see over here, these holes are basically regions of fibrosis. That's an S2 beat. As you can see over here, yes, fibrosis alone is fibrillatory, but the AFib, but the reentry extinguishes pretty quickly. Now, on top of this, if you alter conditions so that you have fibrosis, and on top of that, you have now decreased conduction velocity in the surrounding atrium, as you can see over here. Now see what happens. You got a double whammy, you get fibrosis, and you've got the slowing conduction. One mechanism for which we think is this increase in CAMKII phospholipid NV 1.5. Now, as you can see over here, we saw this multiple times with different iterations of fibrosis plus the slowing of conduction. Now, as you can see, the reentry tends to perpetuate itself. And so we saw this with a variety of different simulations. Again, fibrosis alone, it's possible but hard to get sustained reentry. Now, if you add some slowing of conduction to the surrounding myocardium, different iterations of fibrosis, as you can see, you can sustain the reentry. And these are some of the electrograms, and they also tend to be pretty disorganized. And so this is again basically showing that if you have inhomogeneous slow conduction, juxtaposed regions of fibrosis that exacerbates conditions for AF. This is the model that we put together. Cellular mechanism was the CAMKII phosphorylation ROS-2, which we think leads to triggered calcium wave and therefore to triggered activity. Whereas via CAMKII phosphorylation of NV 1.5, again upstream, this is downstream of oxidative stress. We think what happens is that there's a patchy dropout of NV 1.5 at the intercalated disc, which leads to inhomogeneous conduction. So again, very quickly now, last couple of minutes, this is the gene therapy part of the oxidative stress. Oxidative stress contributes both the initiation and maintenance of electrical remodeling in AF, with the second part of the hypothesis. So in a rapid atrial pacing model, lately we found that NOX2 generated oxidative stress tends to get increased, kind of what we saw in the heart failure model, but then we also found that the sources of oxidative stress are, well, in this case, we found that NOX2 was a big source and mitochondrial ROS was the second source. This is a money slide. Anna was a joint first author with Shin Yu on this paper. What we found was that when you actually perform targeted expression of NOX2 siRNA in the intact atrium, again, these are, just focus your attention to the right-sided panel. As you can see over here, if you just paste normal dogs, and now we're looking at more than 8 hours of AFib. If you paste normal dogs, after about 4 or 5 weeks of pacing, they'll develop 8 hours of AFib, and then they'll stay in it thereafter. But in animals that received NOX2 siRNA, as you can see over here, there were 6 of them. These animals did not develop AFib for that entire period. And we've actually followed a couple of these animals up to 6 or 7 months. And while they may go into flutter, but they do not go into AFib. Possible mechanism is that we think there's prolongation of refractive periods in these atria. This is just in the presence of NOX2. ERPs tend to be longer. And then at the cellular level, we believe the reason you have this attenuation of ERP shortening is because there is an ion channel called constitutively active IKACH. And so the three, so canonical thinking, we know for years, people have known from Stan Natel's data that L-type calcium channel, a decrease in L-type calcium channel is shown over here. So this is normal atrium, this is rapid pacing, there's a decrease in L-type calcium channel, which for years has been thought to be responsible for refractive period shortening. But we also know from Stan's group and other people's group that there's an increase in IK1 that occurs in these atria, and there's an increase in this constitutively active IKACH channel. So we looked at all three to see which of these would be the most ROS sensitive, and interestingly, L-type calcium channel did not appear to respond very much to ROS inhibition, neither did the IK1, but the constitutively active IKACH was very, very sensitive to ROS inhibition. And so we believe that the reason, and this, we actually saw this, when we actually performed targeted expression of NOX2 siRNA in these atria, we were actually able to uptund this upregulation of this constitutively active IKACH channel. So we think that that's a major cellular mechanism by which this NOX2 siRNA is leading to a prolongation of refractive periods and a decrease in AFib. To summarize, this is the end of the talk. Increased parasympathetic nerve sprouting contributes to formation of a vulnerable AF substrate, both in the presence and absence of structural heart disease. Pro-fibrotic TGF-B signaling in the posterior left atrium underlies adverse atrial remodeling in heart failure. Oxidative injury creates substrate for triggered activity and re-entry into pulmonary veins in heart failure. And NOX2 generated oxidative injury underlies the genesis as well as maintenance of electrical remodeling in AFib. So the clinical implications of this are the gene-based therapies where we can overexpress G-alpha iono and inhibitory peptides, go after TGF-beta signaling, or go after oxidative stress, may allow for the successful targeting of electrical and structural remodeling in AF, and the future optimization of gene-based therapy, gene therapy approaches may translate into an effective therapy for AF. Thank you for your attention. Just wanted to thank people in my lab, my collaborators, as well as funding sources, and thank you for your attention.
That was great, Rishi. You know, obviously very thought-provoking stuff. Maybe I can just ask a couple questions. We're a little over here. So Alex asked a question which we've discussed before, but because the autonomics probably play a very important role, do you think that this new attempt at pulse field ablation will have an Achilles heel if it doesn't target that? Or as you kind of alluded to in your statement, do you think it's not as selective as people are saying?
Yeah, I think the latter. I really do think electroporation, at least in our hands, is fairly agnostic in terms of cell type. So as long as you get nerve fibers and yet you don't get, so the question is, does it spare certain cell types or does it spare certain structures because they're far away? I think the answer is the former, the latter. I do think in its field, it does get, whichever field you're electroporating at, I do think it gets most cell types under its field, but I think its at a distance effects are the beneficial ones, and that you don't get the collateral damage that you would expect. I think that's what's going on, Dishant. I don't know for sure, but at least if I had to, surmise what might be happening in terms of what the benefits of PFA might be vis-a-vis collateral damage, I would say I do think you get effect on neurons. And I think the reason you do is because the nerves, the nerve fibers are right there. They're actually in your field of electroporation. So I think whatever's in your field, you will get. Whatever's a little bit further away, you probably will not. I would say that's my take on it. Now that being said, when we've done electroporation, we found that we actually do see a far field effect. I don't know how big it is, but we do see that if you have electrodes in close contact with myocardium, you don't need great contact with myocardium to get, and that's what PFA studies, I believe, are saying also. You don't need great contact with myocardium to create the electric field. So I don't know what the perfect answer to that is. So I think you do have some far field effect of PFA, but I think it's a limited far field effect, which is one of the reasons you don't get collateral damage. But I think within your site, but within the region that's actually getting electroporated, I do think you get most cell types. I don't know if that makes sense.
Okay. Well, I mean, we'll probably get more information as more of us get our hands on those catheters. But in the interest of time, I'll just ask one more because, our audience here is mostly the fellows. So as a clinical electrophysiologist, it seems that our recent attempts at anatomic-based or even electrogram-based ablation for AFib have not really moved the needle in terms of success rates over the last few years. So if you had to give your opinion to the upcoming trainees, where would you say that we should focus our efforts, since it doesn't seem like we're doing much in terms of adding to our ablation strategies?
Yeah, that's a great question. So I would say my answer to that would be that you're right. There is this dismay in the field that electrogram-based approaches have failed us. And unfortunately, anatomic approaches have their limitations for the reasons that we talked about. But I do believe that one of the reasons electrogram-based approaches have failed is because we were targeting a phenomenon, namely rotational activity, that may or may not really reflect true substrate. So what I mean by that is, you can see, for example, from some of our own studies, clearly, underlying AF substrate, for example, what I showed you with the autonomic data and fibrosis and what have you, does affect AF electrograms. So I think if you're going to do a mechanism-guided AF ablation, I do think you can target electrograms, but then you should target electrograms based on whether they're really reflecting true pathophysiological AF substrate, instead of going after a phenomenon that may or may not be reflective of that substrate. So I would say For example, if you wanted to target the autonomics, one of the mechanisms, one of the ways by which you could do that potentially from our own data is you could use atropine, or you could do, as some other groups are doing, is do vagal nerve stimulation. But if you were to use atropine and then go after regions or electrograms that organize most in response to atropine and then ablate those, I would say that would be a better way to do mechanism-guided or electrogram-guided ablation than to, again, focus their efforts in finding something that looks like a reentrant phenomenon and then chasing that instead of going after the root cause mechanism of that. I'm not saying rotors don't exist, but I also feel that there are many mechanisms that underlie these rotors, and ultimately, if you're really going to get success, it has to be after going after root cause or fundamental mechanisms. So I do think there's a lot you can still do in terms of using electrograms to better guide ablation, especially in persistence, where really, other than posterior wall isolation and expanded ablation, where there's little else we can offer those patients right now. I think in those patients, we have to seriously think about developing approaches to target electrograms that, again, as I said, are truly true mechanism guided. And as you know, my lab is working on a couple of those lately. So I think there is still a lot that can be done in that area. I just think we started in the right direction, but then we quickly, I think, went down a rabbit hole and decided to go after the electrical phenomenon instead of going after the